Hi, everyone. Thank you for the introduction and thank you for the opportunity to speak here today. My name is Elaine Ho. I'm a research software engineer at the Rosalind Franklin Institute. And my talk today is on fairly depositing biological segmentation data, where we're at, why you should do it, and how we can do it better in the future. So as a research software engineer, a lot of my role is developing software tools for processing microscopy data. And it's really important to be able to validate that my tool works on a big range of data. I often find when I'm looking at publications, I find like the perfect data set to evaluate my tool on. And then I look in the data availability and it's either not available or available only on request. And I'm sure I'm not the only one who has come into that problem before. So I wanted to find out how prevalent this problem is and what we can do better in the volume EM field. My talk is going to consist of a few questions I'm going to answer. First off, what is biological segmentation data? The next one is how fair are we being in the volume EM field over the last 10 years in this space? What are some of the barriers to reusing the segmentation data and what we can do better to improve it, and some recommendations for everyone to take on board. So I'm sure everyone might have come across this acronym before, but FAIR stands for findable, accessible, interoperable, and reusable data. And it's really important for biological segmentation data to be fair because it's expensive and it's really difficult to generate, especially if you're going to do it manually. So to get the best kind of return on your investment in time and effort, we should be making this fair and openly available so the data lives beyond the original study it was intended for. The second reason why data should be fair is because we need benchmark data sets. I'm sure everyone has come across many different tools which claim that they do the same thing, but it's really important for us to have benchmark data sets to fairly compare these tools together. And the third reason why segmentation data should be fair is because AI approaches, which help us reduce the amount of manual work we need to do, these require high quality training data, and it needs to be suited for the segmentation problem at hand. Most of the time, a lot of the AI tools might be developed by groups of computer scientists who might not have access to a multi-million-pound microscope and the expertise to use it. So it's important for us to be able to share all of these data that we collect to be able to enable lots of other people in different fields to contribute and make our lives a little bit easier. So we were looking at a literature review that surveys the field to find out how can we as individuals and a community improve the reuse of biological segmentation data. We looked at the last 10 years of publications in the volume EM field, and there were 227 publications. I found them from a few kind of common databases in the field, as well as a PubMed search for segmentation or annotation in different volume EM techniques. For each of the publications, we studied the publication type. So was it a software paper? Was it a bio paper? Where all of these studies got their data from, and why they did the segmentation. So was it quantitative? Was it qualitative? And also for each study that made data, where did this data end up after it was published, and what file formats were used for it? Just a quick round of definitions for types of the biological segmentation data we looked at. First off is probably the most straightforward, so raw data, which comes from the microscopes. The second category of data we looked at was training data. So if you had an AI model used in the study, were the images and the labels used to train this model made available online? The third type of data, which is the most heterogeneous, is the label data. So generally, this describes the feature of interest. It could be an annotation, it could be a segmentation, and it's usually the data you look at to get your study results. And lastly, we looked at whether the code was available. So these are the software or the scripts that were used to generate the data if you were looking at doing it computationally. So maybe unsurprisingly for some people who might have looked for label data in the past, we found that 82% of the studies we looked at did not deposit their label data fairly. That's 182 studies of the 227 we looked at did not deposit it at all. Considering that a lot of this research is publicly funded, it's quite sad that it's also not publicly available. And when we look at kind of where a lot of the volume EM data studies get their data from, we also see that very few of them reuse data from elsewhere. This is okay because we should be collecting new data and pushing the boundaries of where we can go with it. But also if there's data that's already available for your particular problem, and this is the case for a lot of software papers, we should be trying to reuse data that already exists where it's available. And this kind of tracks low rates of fair data deposition results in low rates of data reuse. So in the next few slides, we're going to look into a few reasons why this might be the case based on the literature review that we were looking at. So the first reason why segmentation data might not be reused is because we can't find it or access it in the first place. These two plots show the deposition locations of raw data that comes off the microscope and label data that was generated in the volume EM field. We find that more than half of them, even in the raw data kit, were not even deposited at all. And in the label data, this is like 82.4% actually, which were not findable or accessible. So of the data that is findable and accessible, the most common place that it ended up was in field-specific databases. So this could be places like Empire, EMDB, or OpenOrganelle. And that's usually kind of the first port of call if you're a software engineer or data scientist trying to find good data sets to work with. Another popular place for data to end up is in general scientific databases. So this is a place like Zenodo, which gives you a DOI for your work, but you wouldn't go to Zenodo to search for a specific data set because you're probably not going to be able to find what you need. So self-maintained websites were also pretty popular, and this is like your lab website or an institutional repository where the responsibility for long-term data storage is on the researcher who put it there in the first place. And the last place that it's common for data to be put in findable and accessible locations is in a public challenge. So for people who might not have seen these before, a public challenge is usually a really curated set of data which is uploaded together with a clearly scoped problem and clear performance metrics that people can participate in and compare different software tools against each other on a benchmark data set. So this is a great way of kind of making really good use of data sets that might be novel or really helpful for a lot of people. The next reason why we found segmentation data was not reused is that we have too many file formats. Across all of the different data types, we found there were 21 different ones, but this is only 227 publications. So there are way more file formats out there than the ones we've looked for. I've grouped these into 4 different categories. So the first, we've got chunk data. And this is kind of where we should be going towards because it enables us to do streaming and online viewing of our big and bigger data sets. We've also got a lot of data hosted in general file formats. So this could be things like TIFFs or PNGs, but zipped files were also really common. So if you're ever going to upload a zipped file, please say in the metadata what format you're going to use because it's quite frustrating to download 100 gigabytes of a zipped file and then find out that you don't have a subscription to the software that's needed to open said zipped file. The next most popular category, I guess, of file formats is microscopy-specific file format. So the most common ones were MRC and MAPS, which are, I think, basically the same thing. But there are lots of other ones out there as well. And proprietary file formats kind of form a fair amount of the training and label data, kind of especially because they might be locked to a specific software. So the kind of conclusion from this slide is that we've got a lot of file formats out there, and we kind of need to agree on which ones to use, or at least have the tools available to convert between each other. Bioformats is one really good one, but it would kind of be nice if we didn't have to do that in the first place. And speaking of consistency, the third main reason we found for why data might not be reused is because we are quite inconsistent with our definitions. It's really hard to find the specific type of data we're looking for. So an example here is that all three images on this slide were described as masks in the database that we kind of looked at. And they all represent very different, they were all generated in very different ways. So the first one could have been a manually annotated part of a membrane that might be interesting. The second one could be a prediction. I don't know if it was manually corrected or not, or how it was made. And the third one, which is maybe the most dangerous one, is that these are particle picks, which for people who from the single particle world or the subtomogram averaging world would be very familiar with, but people who might not have done this before might mistake this as a segmentation of something really small. But particle picks are actually points placed on locations of the center or particles that you want to average. So all of these data are described as mass, and they should kind of be described a little bit more clearly. Which brings me very swiftly on to how we can improve the reuse of our segmentation data. Clearly describe our data and standardize our definitions. And I know this is easier said than done, but there are some language terms that we can use to make our data more descriptive and easier to understand and apply appropriately. So here I've got 4 different types of images that might form part of a biological segmentation data set. And underneath, there are a bunch of words that are used in the literature to describe images that look just like these. You can see that there are some words that are used across different data types and usually not attached with a descriptor, so we know what it kind of looks like. So I'm going to kind of cross out a few words here that are dangerous to use, which are easily misinterpreted. So first we've got reconstruction, which is used in raw data, but also maybe in segmentations. There's also ground truth, which means something very different in AI than it does in microscopy. And the last one, which is I've touched on before, was segmentation. A particle pick is not a segmentation, but sometimes it's described as such, so that can be kind of misleading. The second thing we can do to improve reuse of segmentation data is use public challenges. So we found that if you want to get the most reuse out of a single data set, public challenge is the best idea for it. There was a single challenge held in 2012, which was reused in 17% of all studies which reuse data from elsewhere. That's a single data set that resulted in loads of different publications and loads of software tools that helped solve the same problem that the public challenge was having. And this comes from the connectomics field, where they have very clear performance metrics on how they evaluate their data. And the last thing we can do to improve our reuse is investing in places to store and share our data. Learning from the different volume EM modalities, we find that the EM group has much higher rates of data reuse than X-ray. And they also have much higher rates of depositing data in findable and accessible places. And when we look at the breakdown of where EM data ends up, a lot of it ends up in field-specific databases. So this is like EMDB, Empire, now the CryoET data portal, and loads of other EM-specific things. So I'll stop soon. Yeah, having places to store and share data makes it easier for us to deposit it. I've got some take-home messages here for individuals, the community, and repositories. So basically, deposit your segmentations, describe them clearly, and store them for a long time if you're an individual. As a community, we need to agree with each other and maybe use public challenges to share really good data sets. And on the repository side of things, we need to be developing tools that help developers to programmatically search, access, and process data. So things are more reproducible. Having online viewers helps us to study data before downloading it. And we also need to include bad data and everyday data so that your software tools will work for you every day instead of just on one data set. And thank you for your attention. And thanks to Michelle and Demetrios for a lot of their work on this. And I'm happy to take any questions now.
Thanks a lot. Are there any questions from the audience?
Thank you, Elaine. You know how much I like this work, and thank you for the wake-up call. I have a question. You talk about bad data, and you want to talk about how to describe our data. Is bad data enough for you, or do you want bad data, bad eyes, bad data, bad tilt? What would you look like for bad data?
Ooh, that's a great question. As someone who's not a microscopist, I would actually like more description. So when someone says the data is like a tilt, is bad, for example, it would be great if we had like, I don't know, 1 to 5 score of how much ice is there in this data, or how, or maybe a note that we should not use this tilt and why. So that's a form of annotation that we can use. And so it forms part of this whole data set that we might be able to upload. And that's really helpful in learning what exactly bad looks like. And we also need more descriptive and quantitative measures of it as well. Yeah, thank you.
So especially on this slide, you mentioned this plus VE and minus VE examples. What are those?
Oh, right. Okay. That refers to positive and negative examples. So for example, if you were developing a software tool, I would like to see which examples worked and which examples didn't. So I could assemble like a challenge data set of data that I feel other software tools should work on, but might be challenging. So yeah, there needs to be a bigger discussion, I think, on how much gets archived. But first, we actually need to think about how we measure quality and what counts. What is a good data set to you?
What would be your choice of file format?
Great question. My favorite choice of file format is the one that you do not have to access at all. So something that's stored behind the scenes in a chunks format that you can access through an API would be great. So if I could, for example, write a Python script which says, please download this particular dataset ID at this resolution and this ROI, and it downloads it for me in a nice array that I can interpret and use, that would be great. So I don't even have to interact with the chunks format behind the scenes. Mizar. Oh, Mizar.